Hello. So today we are going to solve the next topic from genetics, and it is a daily practice session. We are solving DBT PYQs, DBT BET PYQs. So let's start with multiple allele and gene interaction. First question: a cross between two true breeding line, one with dark blue flower and one with bright white flower, producing F1 offspring are light blue. So it is a case of incomplete dominance. When the progeny F1 progeny are said 1:2:1, dark blue, 2 light blue, 2 white blue is observed. Yeah. What is the genetic phenomenon is associated with these results? Epistasis, incomplete dominance, codominance, and inbreeding depression. We know it is a typical incomplete dominance. The gene which suppressed by one non-allelic gene without interaction through interaction is known as, okay, so one gene is getting suppressed by another non-allelic gene. This is getting suppressed, and what is this phenomenon is known as epistasis. And the question is asked, the gene which is suppressed, so this gene is getting suppressed, and what we can say to this gene, what we can name to this gene we can name is hypostasis. Epistatic gene is this one who is suppressing another, and the gene is getting suppressed is hypostatic, right? Next question: the genotype of a husband and wife is capital A, sorry, IA and IA. So this is IA and IA. Okay, IA crossing with IA small I. Let's say. Among the blood type of their children, how many different genotypes and phenotypes are possible? So from this genotype, we can get only A blood group and A blood group only. Yeah, we can get only A blood group because there is both allele are with A, and here is A and O blood group. O allele are there, so we can get phenotype A. But if we see the genotype, we can get IA genotype 1 and another IO genotype also. So two genotype we can get, and phenotype we can get is 1. So two genotype and one phenotype are possible. If a man of blood group AB marries with a woman of blood group A, the father whose blood group is O. Okay, so the woman's father is O blood group, and the woman is A blood group. So the woman is actually IAIO. What to and the man is IAIB because he is having AB blood group. What to what different blood type can this man and woman expect their children belong? So from this we can get A blood group, we can get A blood group, this could be B blood group, and this could be AB blood group, and this could be B blood group. So we cannot have, I mean, they cannot have a O blood group child. So they can only consider A, B, and AB. A, B, and AB. Next, in human, pointed eyebrow are dominant over smooth eyebrow, and widow's peak frontal hairline is dominant to continuous hairline. So they are talking about two genes. What phenotypic ratio would you expect in the offspring from a marriage between individual heterozygous for both the genes and individual homozygous recessive for both the genes? So one is heterozygous for both the gene and one is homozygous for both the genes. So we can write it like capital S small a capital B small b crossing with small a small a small b small b. So it is a test cross, and we know if we see a 2-point test cross, the ratio will be 1 is to 1 is to 1 is to 1. Which of the following is incorrect with respect to modification of Mendelian dihybrid? Complementary gene interaction 9 is to 7. That is true. Recessive epistasis 9 is to 3 is to 4. That is also true. Dominant epistasis 12 is to 3 is to 1. That is also true. And additive gene interaction, polymeric interaction 10 is to 6. No, we didn't study anything about that. If a man blood group AB marries to a blood group, the same question I guess. The marriage, the woman of blood group A whose father was to O blood group, what different groups can the man and woman expect in their child? Yeah, this is the same question we have already did. The next, in the complementation test, Benza simultaneously infected E. coli cells with two phages, each which carried a different mutation. Okay, what conclusion did he make when the progeny phages produced normal plaque? The mutation occurred at the same locus. So, if the mutation occurred in same locus, then recombination if happened, then this mutation will remain there. So this can't be an answer. The mutation occurred at different loci. That could be an answer, because if two fudge is getting recombinated, then there is a chance to have a plaque. The mutation occurs close together on the chromosome. If close together and it is saying the same locus are kind of similar, so the gene source cis configuration is the same, is having mutation in the same locus. So only option 2 is the different one. Next, which of the following can have determined if two mutations are allelic? Okay, can help to determine two mutations are allelic. Lack of recombination between two mutations. The two mutants could not complement each other. Suppression of one mutation. Suppression of one mutation by other. Yeah. See, if there is if there is two mutations, there are two mutations. There are two mutations. So how can we get that it is it is allelic or not? If it is allelic, then then let's suppose the previous gene is capital A, and it is getting mutated and having small a. This is allelic. And can one allele suppress another? No. So suppression of one mutation is incorrect. Co-segregation is possible? No. Lack of two mutants do not complement each other. That's true. Small a can't complement another small a. And lack of recombination between two mutations. That's also incorrect. So option 2 is the correct answer. Next, a cross between true breeding line one with dark blue flower and other with bright white flower, produces F1 offspring that is light blue. The same question again, and it is we have already seen that it is a incomplete dominance. Next, in ABO blood group system, human allele IA and IB are codominant and both are dominant to allele small I. If type B with genotype type IB small I woman marries with a genotype A small IA small I, the probable children are obviously all the possible types, that is A, B, O and LE, because let's say A and small I crossing with B and small I, so they can be AB, they can be A, they can be B and they can be O. AB, A, B and O. Right. The gene encoding enzyme A that functions in a metabolic pathway, that conversion of metabolite X to Y knocked out, but still results the formation of Y. So the enzyme is necessary but not sufficient for formation of Y. But if it is necessary, then when A is mutated, then the Y should not be produced. So this is not right. Enzyme is sufficient but not necessary. That's true. It is not necessary. Enzyme is neither necessary nor sufficient. That's also incorrect. And enzyme is both necessary. Enzyme is not necessary. We have already seen. So option 2 is the correct answer. Next, we wish to produce a metabolite X whose biosynthetic pathway is feedback inhibited in the wild type strain. A mutation which is lead to overproduction of X is discovered in the gene coding for homodimorphic enzyme which catalyzes the first step of the biosynthetic pathway of X. The mutation most likely occurs. And the catabolic active site, regulatory site, the of point between contact of two monomer and it is in the random coil of the protein. So the first they are catalyzes the first step in the biosynthetic pathway. So the first step obviously will be the activation of regulatory site, or we can stop the regulatory function of that site. So that should be regulatory site. Next, a cross between two black Labrador retriever. The phenotypic ratio is 9 black puppies to 3 chocolate to 4 yellow puppies. So the ratio is 9 is to 3 is to 4. And this is an example of epistasis. We have seen that this is an example of recessive epistasis. The term co-suppression was coined by Richard Johnson to explain, okay, patchy flower. Co-suppression is kind of similar to co-expression, and co-expression is co-dominance. So co-suppression is also known as, also you can consider as co-dominance. So patchy flower we can see in co-dominance, it is true. Resistance of viral infection in tobacco. How that character, how could be example of co-dominance? That is incorrect. Production of white flower, that is also incorrect. Silence of actin gene, that is also incorrect. That also cannot be example of codominance. So only codominance example is patchy flower. A plant with orange flower was self-pollinated in F1 progeny. We obtained 38 progeny producing red flower, 38 progeny producing red flower, 80 plants producing orange flower. So 80 plants producing orange flower and 41 plants producing yellow flower. 41 plant producing yellow flower. So this is a ratio of 1 is to 2 is to 1, red is to orange is to yellow. The gene flower can shows incomplete dominance. That's very true. The gene color is polygenic trait. No, we have seen that this kind of phenotype we only found in incomplete dominance. The gene flower color is epistatic. No, the trait flower is maternally inherited. No. So this is for today. We have seen all the questions from the topic multiple allele gene interaction. So we'll do the next from human genetics on the next day. Thanks for watching.